For as long as people have looked closely at faces, they have noticed that eyelids do not all behave the same way. Some eyes show a crisp fold of skin sitting neatly above the lash line, creating a visible crease that separates the brow bone from the eye itself. Others show something different, a smooth, unbroken sweep of skin that drapes down from the brow and settles directly over the lash line, hiding the crease entirely or nearly so. This second pattern is what most people mean when they say hooded eyes, and for a very long time it was treated as little more than a cosmetic curiosity, the kind of thing makeup tutorials talk about and eyelid surgeons make a living correcting, but not the kind of thing that seemed to carry any deeper scientific weight. That assumption has not held up. Over the past decade and a half, a slow accumulation of population genetics research, developmental biology, and comparative anatomy has converged on a much more interesting and much more specific story about where hooded eyelids actually come from, why they exist in some populations far more than others, and what deep evolutionary pressure appears to have shaped them in the first place. The story that has emerged is not simply about appearance. It is about climate, migration, ancient survival, and a single, remarkably well-documented genetic variant that geneticists consider one of the strongest and most unambiguous signals of recent human evolution ever mapped in the human genome. What makes this particular story stand out from the much larger and murkier field of trait genetics, the kind of loosely supported claims about single genes controlling complex features that populate consumer DNA, test results, and casual internet science writing, is how unusually solid the evidence actually is. Most physical traits that get attributed to a gene in popular science coverage are, on closer inspection, governed by dozens or even hundreds of genetic variants acting together in ways researchers are still working to fully map. Eyelid shape, as this article will get into, turns out to be a rare case where one variant in one gene does an outsized amount of the work, and where that variant's evolutionary history can be reconstructed with a level of precision usually reserved for textbook examples of natural selection rather than everyday physical traits people compare with their siblings in the mirror. To understand why this matters, and why the genetics turned out to be so much more precise than anyone studying eyelids expected, you have to start with the anatomy itself, because hooded eyes is actually a term that gets used loosely to describe two related but biologically distinct features, and untangling them is the first step toward understanding what the DNA evidence actually shows. The first and more clearly defined feature is the epicanthic fold, sometimes called the epicanthal fold, a fold of skin that arises from the upper eyelid and extends down to cover the inner corner of the eye near the tear duct. This is a specific, anatomically discrete structure, and it is extremely common in populations with East Asian ancestry, indigenous ancestry across the Americas, and in several Central Asian and Siberian populations, as well as in the Sami people of northern Scandinavia, and less frequently in certain African populations, including some Khoisan groups. It is present at birth, it does not depend on aging or skin laxity, and its distribution across the globe is not random. It clusters, geographically and genetically, in populations whose ancestry traces back to specific regions of Northeast Asia during the last ice age. The second feature, which is what most Western cosmetic and beauty discourse actually means when it uses the phrase hooded eyes, is different. It refers to an excess or overhang of skin from the brow bone down over the entire upper eyelid crease, obscuring the crease from view when the eyes are open, regardless of ethnicity. This form of hooding can appear in people of any ancestral background. It tends to become more pronounced with age as skin loses elasticity and the brow bone's soft tissue. Support changes and is influenced heavily by the depth of the eye socket and the position of the brow relative to the eye, a set of skeletal and soft tissue factors that are heritable but operate through a broader and less singular genetic pathway than the epicanthic fold does. Both forms are genuinely heritable. Both run in families with striking consistency, but only one of them, the epicanthic fold and the broader complex of East Asian and Indigenous American eyelid morphology it belongs to, has been traced with real scientific precision to a specific gene, a specific mutation, and a specific evolutionary moment in deep human history. And it is that second story, the one grounded in hard population genetics rather than cosmetic taxonomy, that has produced the findings now reshaping how scientists think about hooded eyelids altogether. Before getting to that gene, it is worth understanding just how long researchers struggled to even properly categorize what they were looking at. 19th and early 20th century anatomists documented the epicanthic fold extensively, but almost entirely through the distorting lens of racy owl typology that dominated physical anthropology at the time, cataloging it as a fixed racial marker rather than investigating it as a specific traceable developmental feature with its own independent genetic and evolutionary story. That framework did lasting damage to the field's credibility and, more importantly, produced almost no real biological understanding of the trait itself. It would take the emergence of modern molecular genetics, more than a century later, to finally separate the actual biology of the epicanthic fold from the racial categorization, schemes that had obscured it for so long, and to ask the more useful question, not which race has this trait, but which gene builds it, and why did that gene spread the way it did? The gene at the center of this story is called EDAR, short for ectodisplasin A receptor, and its role in human evolution is one of the more remarkable and well-corroborated chapters in recent population genetics. EDAR is not, on its own, an eyelid gene in any narrow sense. It is a much broader developmental gene that governs the growth and patterning of a wide range of ectodermal structures, the tissues that give rise to hair, skin, teeth, sweat glands, and, as later research demonstrated, the fat pad architecture of the eyelid itself. EDR belongs to a signaling pathway, the ectodisplasin A pathway, that plays a similar architectural role across many vertebrate species, and disruptions to it in humans are associated with a group of inherited conditions called ectodermal dysplasias, which affect the development of hair, teeth, nails, and sweat glands together. A strong early clue, long before anyone connected it to eyelids, that this particular gene had unusually broad reach across multiple visible physical traits at once. A single-point mutation in this gene, identified in the scientific literature as the 370A variant, arose in a human population in what is now central China, at some point estimated to be around 30,000 years ago, give or take, a substantial margin depending on the dating method applied. The mutation itself is a single amino acid substitution, a change from valine to alanine at position 370 of the EDR protein, the kind of small singular molecular event that, in the overwhelming majority of cases across the genome, produces no noticeable effect at all. This is one of the reasons the EDAR story is so scientifically compelling. An almost trivial seeming change at the molecular level turned out to cascade into a strikingly wide set of visible physical consequences, once it began interacting with the broader developmental pathway EDAR governs. What makes the EDAR 370A variant so significant to population geneticists is not merely that it exists, but how fast and how completely it spread. Analysis of genetic diversity surrounding the mutation shows the unmistakable signature of what evolutionary biologists call a selective sweep, a pattern in which a single new genetic variant confers enough of a survival or reproductive advantage that it spreads through a population far faster than random genetic drift could ever account for, dragging along with it the surrounding stretch of DNA and effectively erasing the genetic variation that existed at that location before the sweep began. Today, the 370A variant is found in the overwhelming majority of individuals with East Asian and Indigenous American ancestry, and it is close to entirely absent in populations of West African and most European ancestry, whose lineages diverged from the population in which the mutation occurred before it arose. This alone tells geneticists something important. The mutation happened after modern humans had already spread out of Africa and separated into the ancestral populations that would eventually become East Asians, Europeans, and the peoples who crossed into the Americas via the Beringian land bridge, meaning the trait it produces is a genuinely recent addition to the human genetic toolkit, evolutionarily speaking, not a deep ancestral feature, shared broadly across humanity. The pivotal research connecting EDAR directly to eyelid and eye region anatomy came out of a set of studies led by geneticist Yana Kambarov, working alongside Pardis Sabeti and colleagues, published in the early 2010s, in the journal Cell and in a companion paper detailing the functional mouse work. Rather than relying solely on statistical correlation between the gene variant and observed traits in human populations, a method that can suggest a relationship but struggles to prove that a specific gene actually causes a specific physical outcome, the research team engineered mice to carry the human EDAR 370A variant and then observed what changed in their physical development. The results were unambiguous. Mice carrying the variant showed thicker hair shafts, increased numbers of sweat glands, changes in mammary gland branching, and notably for this story, measurable changes in the fat pad structure around the eye region and the architecture of the eyelid itself, consistent with the anatomical features associated with epicanthic folds in human populations carrying the same variant. The mammary gland finding, while it may seem unrelated at first glance, turned out to be scientifically significant in its own right, adding to a separate and still developing area of research into whether the EDAR variant may have had additional, less visible physiological effects beyond the externally visible traits that first drew researchers' attention to it. This was a genuinely significant scientific moment because it moved the EDAR eyelid connection from correlation to demonstrated causal mechanism. The gene does not simply happen to appear more often in populations that also happen to have epicanthic folds. It actively shapes the developmental biology of the eyelid region through its influence on fat pad distribution and eyelid tissue architecture, in addition to its well-established effects on hair and sweat gland density. The same genetic switch that gave East Asian and Indigenous American populations their characteristically thicker hair shafts also, through shared developmental pathways, shaped the eyelid fold. Which raises the obvious and much more interesting question. Why would natural selection favor this particular combination of traits so strongly and so recently that it swept through an entire ancestral population in what is, on an evolutionary timescale, the blink of an eye? The leading hypothesis, and the one that has accumulated the most supporting evidence across multiple independent lines of research, centers on climate, specifically the extreme cold, dry, and often intensely bright environmental conditions of Ice Age, Northeast Asia, and the Beringian steppe tundra that connected Siberia to the Americas during the last glacial period. This was not merely a cold environment in the ordinary sense. It was a landscape of extreme temperature swings, relentless wind, low humidity, and, especially in winter and spring, blinding light reflected off vast expanses of snow and ice. Conditions that place serious physiological strain on exposed skin, sweat regulation, and the eyes themselves. Paleoclimatological reconstructions of the Beringian land bridge during the peak of the last glacial period describe an environment sometimes referred to as the mammoth steppe, a cold, arid, and largely treeless grassland ecosystem that supported large Ice Age megafauna and the human populations that hunted them, but that offered virtually no natural shelter from wind, cold, or the intense glare produced by low-angle sunlight reflecting across open snow and ice for much of the year. Several of the traits linked to the EDR370A variant map neatly onto plausible adaptive responses to exactly this kind of environment. Thicker hair shafts and increased sweat gland density both plausibly relate to thermoregulation in cold, dry conditions, where retaining body heat while still managing moisture loss during physical exertion is a genuine survival challenge. And the eyelid changes fit a similarly coherent adaptive story. Additional fat padding and a more pronounced skin fold over the eye region would have provided extra insulation for the delicate tissue around the eyes in extreme cold, while also reducing the surface area of eye exposed to wind, airborne ice particles, and the intense glare of sunlight reflected off snow. This is sometimes referred to informally among researchers as the cold adaptation hypothesis for epicanthic fold evolution. And while it remains, appropriately, a hypothesis rather than an absolutely settled conclusion, the deep evolutionary past does not allow for direct experimental confirmation of exactly which pressures mattered most. It is currently the best supported explanation available, consistent with the geographic origin of the mutation, the timing of its spread, the specific cluster of traits it produces, and the environmental conditions the ancestral population carrying it would actually have faced. It is worth noting that this cold adaptation framework for the epicanthic fold, specifically, is a more recent and more rigorously supported successor to older, less well-evidenced anatomical hypotheses that circulated for much of the 20th century, some of which speculated more loosely about protective functions against sand, dust, or general environmental exposure without the benefit of the genetic dating and functional validation evidence available today. The current hypothesis is considered stronger precisely because it does not rest on anatomical speculation alone, but integrates the genetic dating of the mutation, its geographic origin, its documented functional effects in model organisms, an independent paleoclimatological reconstruction of the specific environment its earliest carriers would have inhabited. Researchers studying selective sweeps of this kind also point out that ADAR's broad reach across multiple ectodermal tissues may itself help explain why it was able to spread so quickly and so completely. A genetic variant that offers only a marginal advantage in one narrow area of physiology often struggles to become fixed across an entire population within a relatively short evolutionary window, because the selective pressure in any one specific direction may not be strong or consistent enough. A variant that simultaneously improves thermoregulation through hair and sweat gland changes, offers ocular protection in extreme glare and wind, and does so all at once through a single coordinated developmental switch, would have represented an unusually efficient evolutionary package. Multiple small survival advantages bundled into a single trait, compounding across generations of intense environmental pressure into the kind of rapid, near-total sweep the genetic record actually shows. What makes this story more than an interesting footnote in evolutionary biology is what it means for the roughly billions of people alive today who carry this variant as part of their ancestry. A population that spans East Asia, Southeast Asia, Central Asia, Siberia, the Arctic, and the entirety of the indigenous peoples of North, Central, and South America, whose ancestors carried the variant with them across Beringia into a hemisphere no modern human had previously inhabited. The epicanthic fold, in other words, is not a minor or incidental cosmetic feature scattered somewhat randomly across humanity. It is a direct, physically visible, and genetically traceable legacy of one of the most demanding environmental crossings in the entire span of human prehistory, carried forward, quite literally written into the eyelids, by descendants who have no memory of the Ice Age, step their ancestors survived, but who carry its evolutionary signature on their faces every day. It is worth pausing here to address something the genetics makes clear, but that popular discussion of hooded eyes often gets muddled. Not everyone with a visually hooded eyelid appearance carries the EDR370A variant, and not everyone who carries the variant has a strongly visible epicanthic fold. Genetics rarely produces perfectly uniform outcomes. An eyelid appearance specifically is influenced by a combination of factors beyond any single gene, including the general category of hooding driven by brow position and orbital skeletal structure discussed earlier, which operates largely independently of the EDAR pathway and can appear in any population regardless of ancestry. The genetics gives us a real, causally demonstrated mechanism for one major and clearly defined category of hooded eyelid, the epicanthic fold complex, associated with East Asian and indigenous American ancestry, while leaving the broader, more anatomically diffuse category of age-related and brow-driven hooding as a genuinely separate phenomenon with its own, less singular genetic architecture, likely involving many genes of smaller individual effect working together with developmental and connective tissue factors, rather than one dominant variant. This distinction matters enormously for anyone using consumer DNA ancestry testing services and encountering trait reports about eyelid type, because it explains an apparent inconsistency that a lot of people notice and find confusing. Two full siblings, both raised in the same family with what looks to a casual observer like similar ancestry, can show noticeably different eyelid appearances, because the specific combination of alleles, or each sibling inherited at the EDAR locus, and at the various other loci contributing to orbital and brow anatomy is not identical, even between siblings who share the bulk of their genetic ancestry. Traits governed by a strong single-gene selective sweep like EDAR tend to be more consistently inherited within closely related family lines than traits governed by the more diffuse multi-gene architecture underlying general brow-driven hooding, which helps explain why epicanthic folds tend to run in families with more predictable consistency than the broader cosmetic category of hooded eyes does. Some consumer genetics companies have, in recent years, begun including simplified versions of eyelid trait predictions in their trait reports, drawing directly on the EDAR association data, though researchers caution that these consumer reports typically simplify a more complicated underlying genetic picture, and that predicted trait outcomes from limited SNP panels should be treated as probabilistic tendencies rather than definitive statements about how any individual's eyelids will actually appear. The research has also opened up a genuinely interesting secondary line of inquiry into the cultural and social history surrounding eyelid appearance, particularly in East Asia, where cosmetic surgery to create a more visible, Western-style eyelid crease, commonly known as double eyelid surgery or blepharoplasty, has for decades been among the most frequently performed cosmetic procedures in countries including South Korea, Japan, and China. The genetics research does not make any claims whatsoever about beauty standards or the social pressures that drive individual decisions about cosmetic surgery, which are shaped by complex and often deeply personal cultural, historical, and individual factors that population genetics has no authority to adjudicate. But understanding that the underlying eyelid architecture being altered by these procedures traces back to a specific, ancient, environmentally adaptive genetic variant rather than being an arbitrary or meaningless anatomical difference, has added a layer of scientific and historical context to conversations about the trait that previously existed almost entirely in the domain of cosmetic and cultural discourse, without much reference to the deep evolutionary history underlying it. It is also worth noting that within East Asian populations themselves, the presence or absence of a visible double eyelid crease varies considerably, since not everyone within these populations carries the same combination of EDAR-associated and non-EDAR-associated eyelid genetics, meaning the trait was never a uniform marker even within the populations where the underlying variant is most common, a fact that surgeons specializing in the procedure have long observed anecdotally, and that the genetics research now offers a mechanistic explanation for. There is also a broader lesson embedded in the EDAR story about how much of human physical diversity, generally dismissed as superficial or cosmetically incidental, actually turns out to be traceable to specific, identifiable, and evolutionarily meaningful genetic events once researchers look closely enough and with the right tools. For most of the history of physical anthropology, traits like eyelid shape, hair texture, and skin tone were cataloged and described, often in deeply flawed and racially essentialist frameworks, without any real understanding of the specific genetic mechanisms producing them or the specific evolutionary pressures that shaped those mechanisms over time. Modern population genetics, working with tools that simply did not exist even 20 years ago, genome-wide association studies, ancient DNA sequencing, functional validation through model organisms, like the EDR mouse studies, has begun replacing that older, purely descriptive approach with a genuinely mechanistic and evolutionarily grounded understanding of where visible, human physical variation actually comes from, and the eyelid research is one of the clearest and most complete examples of that shift currently available in the scientific literature. This shift matters beyond the specific case of eyelids, because it reflects a broader transformation happening across the entire field of human evolutionary genetics. Traits once treated as fixed, largely unexplained, markers of population difference, the kind of features that historically got folded uncritically into crude racial categorization schemes, are increasingly being resolved into specific, well-understood stories about ancient environmental pressure acting on identifiable genes. It is also worth noting, because the genetics research is careful and precise about this even when popular discussion of it is not, that carrying the EDR 370A variant is not remotely equivalent to a simple ethnic or racial category, and researchers are consistently careful to frame their findings in terms of population, frequency, and ancestry rather than discrete racial classification. Human populations are not neatly bounded genetic categories. They are the product of continuous migration, mixing, and gene flow across tens of thousands of years, and any individual's specific combination of ancestral variants reflects the actual, often quite complicated migratory history of their specific ancestors, rather than membership in any clean-edged category. The genetics tells a story about deep population history and adaptive pressure. It does not and cannot tell a simple story about any individual person's identity or ancestry in isolation from the rest of their specific and often quite complicated family history. Where the research goes from here is, according to researchers working in this area, likely to focus on two main directions. The first is a more precise mapping of exactly how many additional genetic loci, beyond EDAR, contribute smaller effects to the overall range of eyelid and orbital anatomy observed across human populations. Because while EDAR's role in the epicanthic fold specifically is now about as well established as any single gene trait association in human evolutionary, genetics gets the broader spectrum of eyelid appearance almost certainly involves additional genes of smaller individual effect that current research has not yet fully characterized, particularly for the brow-driven hooding pattern that appears across all ancestral backgrounds. The second direction involves deeper ancient DNA analysis of skeletal remains from Ice Age Siberia and Beringia, where researchers are hoping to trace the precise geographic and temporal spread of the EDAR variant with much finer resolution than currently exists, potentially pinning down not just the general region and rough time period in which the mutation first appeared and began sweeping through the ancestral population, but the specific migratory routes and population bottlenecks through which it spread on its way toward becoming one of the most successful single-gene adaptations documented anywhere in recent human evolutionary history. What began as a question that seemed almost too trivial for serious scientific attention, why do some people's eyelids look different from others, has turned out to be a genuine window into one of the most demanding and consequential migrations in the entire span of human prehistory, the movement of ancestral populations across the brutal wind-scoured landscape of Ice Age Siberia and the Beringian land bridge into a hemisphere no human being had ever set foot in before. The fold of skin above millions of people's eyes today is not an arbitrary accident of anatomy. It is a physical, heritable, and now genetically documented record of exactly how those ancestors survived an environment that tested the absolute limits of what the human body could endure, encoded not in any written history, but in the eyelids of their descendants, carried forward silently across hundreds of generations until modern genetics finally learned how to read what was written there all along. And in the way that genuinely well-supported scientific stories often do, this one closes with more open questions than it answers definitively. Not because the core finding is in doubt, but because a single, clearly documented adaptive event like the EDAR sweep tends to illuminate just how much of the rest of human physical variation remains to be explained with the same level of rigor. If a fold of skin above the eye can be traced this precisely to a single ancient mutation, a specific ancestral population, and a specific set of brutal environmental pressures endured tens of thousands of years ago, it raises an obvious question about how many other features of the human face and body, still cataloged today as simple, unremarkable, variation, are quietly carrying their own unwritten histories of ancient survival, waiting for the right combination of genetic tools and scientific curiosity to finally bring them to light.